So now let's have a look at bioinformatics pathway. Personally, I experienced a rapid learning curve last summer as I ramped up my bioinformatics skills just in time for the start of the session. I had the necessary biology knowledge, but no prior coding experience in OR. So after a series of crash courses, Eve's STEM-away tutorials, and nagging my brother, who is very skilled at coding, I was ready to become project manager of the bioinformatics pathway. This also allowed Debelina to notice the potential for pathway hubs. So I encourage you to join these as soon as possible. And the good news is you'll have more than 2 weeks to learn an entirely new coding language. So bioinformatics is where biology and information technology meet together, and how one can enhance the other field. Our chosen pipeline for our internships last summer consisted of 3 main parts: normalization and quality control, differential gene expression analysis, and finally functional analysis, where we looked at the real-life applications of our work. I'm going to have a quick overview over the project management side of the bioinformatics pathway, which was where I was leading. So we had 3 teams within the overall pathway. Each team consisted of 10 to 15 participants. So you had the project lead in each team. These were the people responsible for the assigning the project goals. They worked very closely with our STEM mentors and STEM professionals to deliver the instructions to our participants and interns. And then you also had a technical lead in each sub-team. And these technical leads were in charge of giving you help with, say, the OR programming or any coding issues that you may have run into. These people were very skilled in the OR language, and also had a deep understanding of the bioinformatics project. Each team then had 3 sub-teams, and these sub-teams were the people that you could closely collaborate with. So we put 1 task lead each week so that you could put greater emphasis on your strong points and you could lead your fellow team members. So the sub-teams then consisted of 3 to 5 participants, and they worked very closely together with video calls throughout the week to help each other fulfill their goals. So without further ado, I will pass you on to Disha, who will be speaking to you about quality control and differential gene expression analysis. Over to you, Disha.
Hi, I'm Disha. I'm a junior in high school, and I was a part of the June Bioinformatics session this year. I'd like to just start off by thanking my leads, Erin, Isha, and Eves, for an incredible experience and for the help getting started in bioinformatics. In my section of the presentation, I'll be focusing on the quality control and differential gene expression analysis steps of the pipeline, which we learned through recreating the results of a scientific paper. So one of the first trainings we had was about how to read and understand a scientific paper. As an example, in what order should you try and read a paper? Skim the abstract, read the introduction, results, and conclusion, and then go back and read the abstract again. I found that this method of reading the paper was really helpful for comprehending the challenging material found in the paper. We also learned about the GEO database, which is a repository of curated gene expression data, and NCBI PubMed, which has the latest bioinformatics papers. You have free access to both of these NCBI sites, so they're a great resource for anyone interested in bioinformatics. Moving on to the project itself, the aim of this paper, which we used, was to find prognostic markers for colorectal cancer. Prognostic markers, by the way, are biological characteristics that are indicative of the likely health outcomes of a patient, such as disease recurrence. I will explain the required steps of the pipeline and the R-Bio conductor packages involved in the next few slides. But first, let's talk a little bit about the process of generating gene expression data. So how do we get this data? We use microarray sequencing, where an experiment with a single chip or array can provide information on thousands of genes simultaneously. Each dot that you see in the image is referred to as a probe, and it represents a specific genetic sequence. In microarray expression analysis, the sample has genes from both the normal as well as the diseased tissues, which are distinguished using dyes. The brightness intensity of a probe after hybridization denotes the gene expression levels. And that light intensity is converted to a numerical value, and that is the data which we will be examining. Now, in quality control, we have two main goals. One is to check the reliability of the microarray sequencing, and two is to identify and remove outlier samples, which may be caused by biases in the experiment. The RLE and news plots, which are shown on the right, are from the AFVPLM package for normalization. Let's focus on the RLE plot for right now. So most of our data will have similar expression levels. So if we can isolate probe sets that appear to have significantly different expression levels, we can eliminate possible outliers. If you look at the RLE plot, you will clearly see that the last probe set is an outlier compared to the other samples. So there are many quality control packages as listed under the main tools section, and it's a good idea to use multiple packages in order to get a high level of quality control, because some plots may miss certain outliers. Personally, I like the AFVQC report package the best, as it provides several helpful plots within just one function. Now that we've cleaned up the data, we are ready to start DGE analysis. First off, what is an expression level and why is it important? Expression levels are the rate at which a gene is transcribed. So if a gene is involved in a disease, you may see high expression levels called an upregulated gene or low expression levels called a downregulated gene. If the gene isn't involved in the disease, you shouldn't see any variation in the expression levels between a healthy and diseased sample. We did DGE in 3 steps: annotation, gene filtration, and lemma analysis. Annotation is simply linking the biological information to the numerical data. Gene filtration is removing a large part of the data so that only the most significantly differentially expressed genes remain. And in lemma, we fit a linear model to the expression data for each gene. So there's a lot of intriguing math involved in DGE analysis. We don't need to understand all of it right away, as it does get rather complicated, but I would just like to cover 2 key concepts: p-value and fold change. Here's a demonstration of these concepts using a volcano plot. A volcano plot is a type of scatter plot that shows statistical significance, or p-value, versus the magnitude of change, or fold change. It enables quick visual identification of genes with large fold changes that are also statistically significant. These are likely the most biologically significant genes. As an example, the highlighted gene HOMER1 is downregulated and statistically significant, so it is a gene of interest in our research. So returning to the paper we started with, shown on the right, some of this may begin to look a little bit more familiar to you and make a little bit more sense. As you can see at the top of image A, the researchers started with 3 of our data sets from GEO. Then in image B, they identified the upregulated and downregulated mRNAs. Images C and D are the visual representations of what was found, with image C being a type of volcano plot like we discussed earlier, and image D being a heat map, which is a plot that shows correlation between different genes. On the left, I attached a snippet of our code that we used to perform the steps needed to recreate these graphs. And Dan will begin to tell you more about the next step, which is functional analysis. So overall, I faced significant challenges in this internship. For one, because I came in with less understanding of biology than used in the paper. I initially struggled with understanding the bigger picture. And also because I was new to programming, I had a hard time with the different object types and figuring out how to manipulate them. And I think my major achievement in the session was overcoming both of these challenges. Also, working with other people of different experience levels who also enjoy bioinformatics was a really great experience, and I would like to thank the leads and STEMaway once again. I hope this session was informative for future students, and if you have any questions, please feel free to leave them in the Q&A chat, and we will answer them after Dan's part of the presentation.
Thank you, Disha. We'll now hand it over to Dan Binderman, who took part in the second session, the July session last summer, and he will speak to us about the third part, the functional analysis.
Okay, so hello everyone. My name is Dan, and I am a senior in high school. And yes, I'm gonna get right into what I did in the bioinformatics pathway, and is likely what it will resonate a bit what you will do in if you do pursue this internship. So basically what we were doing is that we were looking at the data collected from the report and comparing the data from cancer cells and normal cells, right? And so we looked at the gene expression levels. And so the genes that were expressed really differently in cancer cells versus these normal cells are what we may look at as potentially being significant. Okay, so once we have identified these top deregulated genes throughout through using differential gene analysis, we can now give them more applicable biological meaning through functional analysis. And so in this step, we identify the most common cellular processes related to these genes and identify the proteins that these genes actually code for. Okay, yeah, so I don't want to spend too much time going over this slide, but again, it is important to note that some of the external databases that we did use to perform the this multi-analysis project. So just with a quick show of hands, has anybody here ever heard of the enrich-R, Metascape, or David databases? Let's see if anyone can raise their hand if they have. Yeah, okay, I see one. Okay, yeah, okay, so yeah, so not many of you have, and that's perfectly fine. When I first did this internship, I had no clue what these databases were. I've never heard of them, and I've never seen these icons, these logos, but as I did this internship, I did start to understand what they were and actually how to use the information that they store to do the analysis. So what each of these databases do is that they provide a comprehensive gene set enrichment and annotation tools. And so we use the information that they store to do the analysis. So throughout the course of this bioinformatics pipeline, we use many different tools for functional analysis, and the first of which was gene ontology, using the enrich geo function in R, the programming language we use. So gene ontology uses a comprehensive model of biological systems to identify the top biological processes and cellular components and cellular functions associated with certain genes. And now, while it is important to note that the deregulated genes are not the only indications of how a disease is progressing, the imbalance between upregulated genes and downregulated genes, which Disha did talk about, they do help shed some light on the progression of a disease. So as with colorectal cancer, which is the specific disease that we focused on in our project, so using statistical inferences, we could see that many genes that were more highly expressed in the cancer cells versus the normal cells coded for proteins that had to do with RNA metabolic processes. And now, so let's think, let's go back to our high school biology notes and let's see if this actually makes sense. So we know that a characteristic of cancer cells is that they divide uncontrollably, right? And so a big component of DNA replication is that is RNA. Either it plays a direct role through RNA primers or indirectly, simply by allowing for the construction of different proteins that help in DNA replication. So this is what I mean by giving, this is what we mean by giving biological application to our results through functional analysis. We want to see if it actually makes sense for the upregulated genes to code for RNA metabolic processes. Okay, so the next step of our functional analysis was to use the string database. And what we hoped to get out of this database was to identify targetable genes based on protein-protein interactions. So this is again another database that when I entered this internship I have never heard of, but it really is fascinating with all the data that it stores and how applicable that data is for our project and many other real-world projects. So what we did is that we took our top 100 deregulated, dysregulated genes, and the string database automatically created a map out of them. And the output was this map that connects these genes based on the proteins that they code for. And it kind of gives us an idea of the protein-protein interactions among these different genes. And now I have a close-up here of a specific gene within this map that the string database created for us. And this is the RHOA gene. And so as we see, if we count, it has 1, 2, 3, 4, 5, 6, 7, 8, 9, 9 interactions with other genes. And so this suggests that the RHOA gene, the protein it codes for, interacts with many other proteins involved with the progression of colorectal cancer. So maybe RHOA, the RHOA gene could be a target for drug discovery, which we'll talk about more later. Okay, and then the last functional analysis segment that I want to talk about is the enrich-R analysis. And that basically allows us to consider transcription factors. And I don't know if you all know what transcription factors are, but they basically regulate the transcription of certain genes into eventually producing proteins. So what we see here is the RHOA gene that we were just looking at, which is actually a hub gene because of how many different genes it's connected to. It is regulated by this transcription factor right here. So what this suggests, the RHOA gene actually codes for a microtubule organization protein. And because this gene was actually upregulated, meaning that it was more highly expressed in the cancer cells versus the normal cells, if we do target the transcription factor and maybe somehow use a drug to prevent the expression of this gene, then it will interfere with microtubule organization and therefore DNA replication, right? Because if any of you have taken a biology class yet, then you know that in metaphase, microtubule organization is extremely important. So if maybe we can interfere with that, then this gene, which is highly expressed in colorectal cancer cells, may no longer be able to produce as well-functioning proteins. Okay, so why does this matter? Why are we actually doing this project? So one thing that STEMaway has really emphasized is that it's not just about getting the results. We have to analyze the data that we get and try to actually understand what's going on. How does it conform to what we already understand, and what new things does it actually teach us? So let's think about it through a biological lens again. Genes, as we know, are the fundamental units that dictate bodily functions by coding for proteins. So we can use the functional analysis to see how these proteins are involved in certain pathways that correlate to the progression of a disease, such as colorectal cancer. And you guys might, in your internship, study the progression of a different disease. And so once we identify these pathways, they become potential targets for effective drug discovery. And this could help reduce the effects of the disease, which is ultimately the goal of everyone, right? We want to try to reduce the effects of a disease like colorectal cancer. I just want to say thank you to STEMaway for this really amazing and enriching opportunity. I had a great time, and I can't wait to build off what I learned throughout this internship. And I hope that all of you in the audience will really consider doing this internship. You're going to learn so much, especially working with older students who have more experience.
And now we'll move on to our audience Q&A. So if you have any questions for Dan or Disha about bioinformatics, feel free to post them now. I will stop the screen share momentarily. So our first question is for Disha. Disha, what have you learned about bioinformatics, or maybe about your soft skills too, that you didn't know coming into this internship?
I think I learned a lot about programming. I really didn't know anything about programming before coming in. So Eve's trainings on R were extremely expanding on my knowledge. Additionally, I think one like huge thing that I learned was the importance of using my resources and talking to people who were more experienced than me. Before, I used to not communicate with people. If I was confused, I would just try to figure things out on my own. But I really learned the importance of doing that because people of higher experience levels than me really helped me in this project. And I think that helped me learn a lot more than I would have like usually.
Thank you very much. I can certainly relate that Yves's tutorial certainly brought me up to speed too. Dan, what did you think about the atmosphere in the team? How did the collaboration work, even though there was such a big age gap and skill level difference?
Yeah, of course. So I have to say that I was part of a group that we collaborate really, really well together. I mean, we're still in touch now. We're friends. And even though it was all virtual, we really get to connect with one another. And I was able to ask them for help. Yeah, again, don't be afraid to ask for help. I mean, like for example, I had two people in my group who were more experienced than me. They were both in college, doing graduate work in this field, and so I asked them questions, of course. One of them was like an expert in R, and so I definitely used his help in keeping up to speed and learning how I can program on my own. And so definitely it was a really fun experience.
I'm glad to hear that our team building activities at the start of every meeting helped to make lifelong friendships, hopefully, or at least connections in the field. Disha, where would you like to further this study? Do you think you might study it in later aspects, or will it kind of enhance your learning of any other scientific fields?
I think after this experience, probably going to go into bioinformatics in the future, and I really hope that I can do my own research in the future. It seems really interesting, and I'd really like to go into specifics in like gene therapy, and I hope that this information that I learned will help me when I do get a higher level of biology education in college. So I want to apply it in that.
I'm sure this experience will certainly stand to you then. So we've a question here. Even if we don't have background information in coding, how can we still apply for this internship? How will we catch up with those who are already experienced with coding? This is where the pathway hubs really come in. I'd encourage you to join them as soon as you can. If you're a high school student, it's really a requirement. If you wish to enter specifically bioinformatics pathway next summer, as it is a challenging pathway, but you are more than up for it if you have the passion. The STEMaway community is blind to age, gender, background, experience. Our goal is the passion. If you have the passion and drive to dive into these areas, you will be able to succeed, and you will be given all the supports you need to succeed in them.